Welcome to New York Bio's Virtual Breakfast Series, a digital program started in 2020, bringing you fireside chats with leaders across the healthcare spectrum. This week's episode features Dr. Raymond Donninger, Senior Director of Commercial Development and Early Development Services at Lonza Biologics.
So good morning, everyone. My name is Jennifer Hawks-Bland, and I'm the CEO of New York Bio. We're thrilled you're joining us for another edition of our virtual breakfast. Today, we have Dr. Raymond Donninger with us from Lonza, and also a special thank you to Lonza, who is our partner in this month's series of breakfast. Let's get to talk about early stage development, contract manufacturing, drug discovery. We have a lot to cover today. But before we delve into both Raymond's background as well as the things he's working on, housekeeping, please, I know you'll have questions for Raymond as we go through the conversation today. So please put those in the chat or the Q&A, and Derek and I will get to them throughout the course of our conversation. With that, I'll kick it over to Derek to start our introduction.
All right. Good morning, Raymond. It is great to see you. We are going to talk about something that I think is really important. You think is really important. You've spent the majority of your career at kind of the transition between discovery and first-in-human clinical trials, which is really a pivotal time for biotech discovery. So I'm excited to dig in here. Before we get going, why don't you tell us a little bit about how you got to where you are and a little bit of your journey to before you got to Lonza? Because you started, interestingly enough, in South Africa doing medical trauma work. So I want to hear about the path from there to biotech and clinical development.
Yeah, sure. Thanks, Derek, and thanks, Jennifer. Pleasure to be here. Yeah, it is a bit of a leap, that Derek. I'm not too sure how many of the folk on the call have any insight in terms of what trauma medicine is like in South Africa, but I can tell you it's quite interesting. To put some perspective on it, at the time I was training, we did have physicians from the UK as well as some other European countries, military arms training with us because of the amount of penetrating trauma and gunshot wounds and things like that we used to see in South Africa. So quite an interesting environment to be working in, and I must be honest, I did thoroughly enjoy that aspect and was considering specializing in trauma and emergency medicine. But I'd always had an interest in drug development. I started off my academic career actually as a scientist and switched to medicine, and had some very, very early exposure to basic research in drug development. And it was always an area that I was very interested in. And I was lucky enough to have an opportunity to move into a biotech company, a small biotech company, but an interesting one that had a product in the market that it actually manufactured in-house. So it had a mammalian cell culture production facility. It also had some bacterial fermentation capabilities, an interesting pipeline in immunology, oncology, and autoimmune diseases. And so it was a fantastic opportunity to go in. I went in to help them with some phase 2 clinical trials, actually. Yeah, wonderful grounding and lovely opportunity to get into the drug development space. Gave me some good exposure into what biotechs go through, and a lot of the uncertainty around funding and trying to raise capital every few years and being on the road to try and interest investors in what you were doing. And like with many biotechs, there was a change in funding ownership, and things became a little bit unsettled, and an opportunity opened up to move back to Europe. I'm originally from the UK, so to move back to the UK and join an international CRO at that time, helping clients in the space transitioning from late discovery through into first in human. So that's sort of what I was doing for a while. Then I skipped out a bit and went and worked in consulting for a bit. And there some of it was development, but a lot of it was actually around the commercial aspects of pharma and life cycle management and things like product launches, looking at phase 3 clinical data and assessing the strength of the data. Got pulled back into clinical pharmacology and ran a clinical pharmacology unit for a few years, mainly focusing on first-in-human clinical trials. then back into consulting for a little bit, and then ended up in Lonza in the early development space.
So you really have seen, I mean, you really have an expertise and exposure to the entire clinical development all the way through launch, which is really unusual, right? Because I find that often people find us a niche and then I mean, that's their focus. But I think that gives you a good breadth of understanding sort of, and probably makes you better able to help clients now understand what they need to be thinking about all the way down the road.
Yeah. It's almost like you should have, yes, I've seen that before on your business card.
Yeah.
Yeah, but I guess, and I'm absolutely certain that that you realize this, and that is that I have got a very, very broad range of experience, but that typically then means that the depth of experience is not necessarily what you would expect from somebody who has a singular focus. I think you're right. Where the advantage lies is I can have a look at a particular development scenario and have an eye on the next step or the ultimate goal. And I think sometimes that is a challenge for certainly some of the small biotechs that I've worked with, to really keep a focus on that end goal. It sounds obvious, but sometimes it's so easy, especially in the early stages, to get enthusiastic and passionate about the science and forget that you're trying to develop a drug to a certain budget and a certain timeline.
Right.
Yeah. So importantly, let's stay on that, right? So what are some of the things that, do you have either, I don't know if it's a protocol or anything like that, but what are some of the things you've seen where that rule has kind of been hammered home for you, right? So have you been in a place where you've actually kind of seen the manifestation of, I'll call them bad decisions, but either just suboptimal decisions, and how important they were to development? Because, one of the challenges I think is that there are people usually that are either, there's late stage folks and there's early stage folks, and there are very few that can actually look at that continuum and say, if you make this chess move here, and that means you have an extra phase 2, or you have to do this, here, and it's not going to be optimized.
Yeah, I think, Derek, the way to look at this, and the way I tend to look at this, is everyone, in my opinion, and again, take it from whence it comes, based on what I said earlier about breadth rather than depth of expertise, really does need to do a target product profile. And this is critical. This is the North Star, right? And it's not a document that you generate on, the ideal characteristics of your drug and then leave. It's something that you should be coming back to all the time as you're generating data. And I've seen on many, many occasions small organizations that have done a form of TPP initially. And it doesn't need to be called a TPP, right? It's just whatever your blueprint is for your drug, your ideal scenario, maybe your ideal, your adequate scenario, if you prefer, if ideal isn't great and you just want, what's going to be good enough. That they've established that and then sort of filed it and said, right, we've done that. Our investors maybe wanted us to do that. We've done that, and that's it. And you say, well, when was the last time you went back to that? And it was like, well, yeah, 2 years ago, 3 years ago, whatever. It needs to be a living document. And I've seen time and time again where organizations have gone beyond where they would have drawn a line in the sand to say, we are getting data that doesn't give us comfort that we're going to have a commercially viable product here, and yet they continue because the science is interesting. And I think often that is quite a challenge. And I do see that I do sometimes see that working with organizations that are perhaps academic spinouts, because there is such passion for that mechanism of action, that particular disease pathway, that the funding helps to generate additional information there, but it's kind of gets stuck in that research phase. And so, being able to pull it out of that and sit down and have a really good conversation about the data and where it's tracking is important. But it's not just that. The other element that I've seen on a few occasions is where clients are developing, or companies are developing a particular product, but not keeping an eye on the competitive landscape. And
Right.
so, I've had companies that I've worked with who are developing, for example, a factor replacement therapy that has a slightly prolonged half-life. So patients are going to need to take it once a week as opposed to twice a week, but not keeping an eye on the fact that actually there's a gene therapy that is in phase 3 clinical trials where one dose and that's it. And you've then got to go back to, I'm not saying you can't progress that product to registration and launch, but you have to have a different way of looking at your target product profile and saying, does this still make commercial sense? Or if it does, how much are we going to need to discount versus what we were planning to do? Are patients really going to think in the age of a gene therapy that being able to give myself an injection or an infusion once a week is still something that I would be prepared to go through? Or which patients are actually the ones I'm interested in? And does that make it such a small segment that it's no longer commercially viable? I think these are some of the elements that I've seen. And just one last point on that. Also, some drug developers in the early space, and it may be because of their concerns around funding, etc. But I do quite frequently see people where risk is black and white, and it's never black and white, right? It's either if I have a risk, I must stop, or if I don't have a risk, I can progress, happily, which isn't necessarily true. And a lot of what we do, certainly now, and what I've done in the past, is to look at where there is potential for risk, understand the risk, and understand how to progress even in the face of particular risk. Now, obviously, there's certain scenarios where you would sit back and say that is a go, a no-go. But there are many scenarios where it's a case of, well, how do we manage this? And in fact, if you look at data that's generated preclinically, there's often a lot of risk. And it's just understanding whether or not that risk is actually real and relevant in the human clinical setting.
Yeah. Well, let's stay there for a second, right? Because let's actually talk about this time period where you have preclinical data and, it's in animal models or, perhaps human tissues or whatever, but now you're actually going to put the drug into a human, right? And, this is an absolutely pivotal time for, any therapeutic. And, walk us through how that kind of decision point has changed or evolved throughout your career. I mean, it'd be really interesting to see what you thought of this earlier in your career and what kind of things have kind of impacted that decision-making, that window now. Because, if we think about this, if we think about getting smarter about development and these evolving target product profiles and things, that decision to go into humans and how you do it is really just about the most pivotal decision that you can make in development.
Absolutely. And it's interesting because I think, over my career, there has been a very significant shift in how we look at and assess preclinical data as it relates, or as it translates, if you like, into potential, human clinical setting. I think probably the biggest difference or biggest change that I've seen is was probably the mid-2000s. I don't know if you remember, but there was the Togenero case where a first-in-human clinical trial unfortunately ended up with serious side effects for the volunteers. And that was really a time when we first started to talk about what is the actual biological activity of the drug, as opposed to what is the no observed adverse effect level in toxicology studies. And I think up until that point, and then, with small molecules and with some of the earlier biologics, the no observed adverse effect level was a reasonable and is a reasonable, assessment to make. But as these products became more human specific, as drugs began to target elements of the disease process, like, for example, the immune system, which could activate cascades and massively amplify a signal, we started to need to understand much more how the drug is actually working at a biological level.
Yeah.
And also, we obviously started to develop the tools to be able to do that assessment.
Yeah.
We started to develop better cell models. We started to build better predictive models, etc. And I think that that combination of, being required to assess the no adverse effect level as well as the minimum anticipated biological effect level, and using both of those to calculate a safe starting dose, especially, as I say, for biologics, was a big shift and is a big shift.
Yeah.
And really forced drug developers to better understand the that biological effect and what that therapeutic window or even biological window, I guess you may call it, may be in terms of transitioning into first in human.
Yeah.
And that sort of first in human risk assessment and what's required for that.
Yeah. And this was 2006. It preceded really the wave of kind of PD-1, CTLA-4 antibodies. I'm going to put a review in the chat here for everybody, and certainly preceded cell therapies. So in a lot of ways, it's almost good that this was a canary in the coal mine. Not that it was good that it had these outcomes, but even just looking briefly at the abstract, this was a phase 1 clinical study, and it was an infusion dose of 500 times less than what they showed was safe in animal studies. So, I mean, really, this was almost like the big flare signal of, if you're going to mess around with agonists in the immune system, you've got to be careful. And, I think we saw a lot of this with cell therapies and CRS and a lot of things that we had to deal with. But, thankfully, we had something like this that, waved a big flag before we basically got to the age of immunotherapy, right? Because, ultimately, you're flipping a lot of switches that, immunotherapy as a field changes like every 10 minutes. We find something else that's new. We find a new cell type. And, the amount that we've learned in that last 15 years has been amazing. But, if you look at this now, and this review has all of the preclinical studies that they did, you probably will read through this review and think that every single thing that they did clinically was a perfectly valid decision at the time for the data that they had.
Absolutely, 100% agree with that. I think, you can't turn around and have a look at those data in the context of that time and say that anything particularly was done incorrectly. And we do have assays nowadays that we can run that can actually predict the likelihood of this sort of thing happening. And that's going back to what I said a bit earlier, where there might be certain risks that you identify that are no-go decisions, right? So you do some of this testing and you see on first exposure of the drug to PBMCs that you've got massive activation, cytokine release, etc. You may elect to move forward with a backup candidate, for example. This is exactly the sort of thing that we're talking about. But I think the other major shifts, certainly from my perspective, and coming in, and this was quite funny actually, when I first started in biotech, which was the early 2000s, and I was working on effectively a biological therapy, but we were also looking at dendritic cell vaccine therapy at that time. So really right in the early days of cell therapy. And I can remember saying to my CEO, who obviously had a huge amount more experience than I did, why are we doing animal testing? Why are we doing any animal testing? It doesn't make any sense. These are not just human specific, but they're human. And obviously, if we're going to use them as an immune activator or an immune modulator, the animals are obviously going to have an immune response to them because they're not animal derived, they're human. And I think
not only that, you're putting them into an inbred mouse strain that has a depressed immune system anyway.
Correct.
Other than that, it's a perfect analogy.
but even if we look at it from a general monoclonal antibody or recombinant protein, and we're doing healthy non-human primate studies, you have to question the relevance of that as well. And I think that is a big shift that I've seen over the 2 decades, I suppose, since I've been in biotech, and that is regulators coming around as well to the notion, to the understanding of the fact that there is a limit to what the normal paradigm of standard toxicology testing can actually tell us. And so, you know, we need other elements around it to help us try and understand what the risk is for humans. Now, you know, I don't, from a regulatory perspective, if I was a regulator, I would be the same, right? My first and biggest priority is the health of the human subjects and the human population. So I'm not going to change what I do very quickly. And I need to have scientific data to support that. But I think, you know, we've seen more recently with some of the FDA changes in terms of, well, I guess the legal framework changes, and we've seen it in Europe as well, where there are pathways now to progress some of these products into first in human testing without any animal testing whatsoever, because it's
They've started to open that door.
Yeah, and that's the other big shift, I think, those two. The one is, you know, really understand the biological effect of the drug, and the second is, you know, understanding that some of these you know, therapeutics are so human specific that you know, the standard, the more usual paradigms may not fit any longer.
Yeah. So help me out with in thinking back, right there, we have, you know, mice with quote unquote human immune systems. We have these tools now that have evolved over the last probably 10 or 15 years. Where are we with those, and how good are they? Right? We could think about organoids, we could think about humanized mice, right? From where you sit in kind of the transition to first in human, how much better than, you know, a regular mouse is, you know, a humanized mouse, and how much does an organoid leave off the table from an actual human patient, right? Where are we in that gray area of, you know, not translatable animal data to kind of perfect non-human human, if that makes sense?
I think we're certainly along the path, but we're far away from that end goal, in my opinion. And there isn't any one perfect assay platform or assay type. And this is one of the challenges for drug developers is understanding, based on the product, the target, the disease, the patient, ultimately, what the best suite, if you like, of studies are to run in the preclinical space in order to you know, make sure that you are identifying potential risk, make sure that you are mitigating that risk, and making sure that you're going into first in human in you know, as optimal a way as possible. And one of the challenges, I guess, is that because these things are suboptimal, you certainly can generate data that can be difficult to explain. So the more artificial you go, the more chances there are that you're going to generate data that you may struggle to explain. And obviously, once you've generated it, it's there. You need to understand it, and you need to explain either whether it has a relevance or it doesn't. And this is a genuine challenge for drug developers, because you don't want to be stuck for years and spending a lot of resource, which you often don't have, trying to explain a finding that you never really wanted to look for in the first place.
Right.
So it is a big challenge. I think, you know, the human situation is so complex, and I'm actually giving it a blanket statement, and that in itself is ridiculous, because every individual will respond differently to a particular therapeutic. It depends on their underlying genetics, it depends on their environment, it depends on their disease, the stage of disease, previous treatments that they've had, other diseases that they may have on top of the disease you're looking to treat them for, because many patients are not, you know, unfortunately don't have just one single underlying pathology. So it is such a diverse and complex scenario that I don't think it's realistic for us to think that we're going to have a model that's going to be, you know, perfectly translatable. There are some very, very good tools now from an in vitro perspective, arguably in some instances, perhaps more useful than the in vivo animal space, to help understand the biological activity of the product and how that may relate more specifically to humans than it does, say, to a rat or a mouse or a non-human primate. But yeah, still far away. I think when I first started in again around about the 2005, somewhere around there, I remember making a very confident prediction that in my lifetime we would no longer have any need for animal testing whatsoever, and that we'd probably be able to use in silico tools and AI tools to do drug development. I think I'll temper that and say that maybe within my children's lifetime, because I think I'm more realistic now. There's still a lot of value to be gained in a lot of the models that are looking at an organism level as opposed to just an organelle or a cell level. But yeah, we've still got a long way to go, I think, although technology moves so quickly that maybe it will still happen in my lifetime.
Yep.
Can we go back to and as everyone who listens frequently knows, I'm like the woman on the street. I am definitely not your scientist. Derek is our science question. So let's go back to something you said earlier about sort of following, like an early stage company, because obviously a lot in our New York ecosystem are earlier stage companies. They sort of they keep following the science despite sort of the signals, right, that they're receiving. Do you think from a business perspective that is happening, that was happening more when sort of capital was free flowing? Or are you seeing a change in the way companies are approaching from a finance perspective, how they conduct and where they go ultimately with the signals that they're seeing?
Yeah, it's a great question, Jennifer. I think, so again, in my experience, biotech funding is a little bit like the CRO or CDMO business. It's cyclical, right? It has ups and downs all the time. There's peaks and troughs in demand, peaks and troughs in funding. So it goes through cycles. And I think you're right. I think when funding is not scarce, typically it's obviously a lot easier to spend money.
It applies to us individually too, right?
Yeah.
I couldn't agree.
Universal rule.
But as funding tightens up, of course, I think the requirements around or the diligence around keeping on track in terms of a particular development path is stricter. But I think one of the changes that I have seen over the last few years is most of the biotechs that we work with now have funding partners that are providing direct resource to them. So helping them with the development, helping them keep on track, helping them ensure that, the science, which is interesting, could happily be a research project for a PhD student, but not for the development team, right? That they need to stay on track with where they're going. So that's definitely something that I think we're seeing a little bit more of, or I'm seeing a little bit more of. So yeah, I think you're absolutely right. As funding is a little bit more constrained, so the eyes looking at where the funding is going and are a little bit more keenly focused.
They have to break out their TPP.
Well, yeah. Sorry, Dave.
I was going to say, so if we're thinking back to kind of TPP and strategic thinking, right? So from your position, how does the emergence of, cell and gene therapies and new modalities basically, throw the old rules out the window, right? I think the rule, gene therapy seems to have bent all of the, if you showed a gene therapy spreadsheet to somebody in 2005 on how it would actually make money, people would be like, you're crazy. There's no way you can get that kind of price, and how the heck are you going to manufacture it, right? So where are we in kind of the product cycle of really understanding what to do with either a gene therapy or cell therapy, and commercially how they're going to work, right? And I realize, Lonza sits right in the middle as probably the most important service provider on the manufacturing side of both of those markets. So what has that kind of done to the overall marketplace of thinking about really the fate of these therapies? Because a go, no go decision on one of those is most companies are based on their lead therapy in those areas.
Yeah. So I think gene therapy, cell therapies, absolutely have a place. In fact, the thing that got me most interested in drug development, if you like, when I was at school, was my thinking that disease must have an underlying genetic factor that allows for a disease to take hold. And that includes infectious diseases. So, I've always been really, really interested in the genetics of disease. And I was, naively at school used to think, well, if you could fix whatever the genetic issue was, then there wouldn't be any disease. But, certainly gene therapies where they are single gene deficiencies are have fantastic potential. I think that's already been shown. I think that the place for, let's call them traditional therapeutics, a monoclonal antibody or a recombinant protein or a drug, a tablet that you need to take on a relatively frequent basis for a chronic condition long term, that market is still going to be there and going to be there well into the future. There are many, many markets in the world that just simply cannot afford cell and gene therapies, and they are also difficult to access from a logistics perspective, right?
Mm-hmm.
So drug developers will have a market for the sort of let's call them, I don't know, traditional drugs. I don't know what we want to call it. Non-advanced therapeutic medicinal products. How about that?
So that's it. Come up with an acronym for that.
Exactly. So I think that's that's going to be the case for many, many years to come. So if I'm developing a drug and I have got a competing cell therapy, for example, I still think there's a significant opportunity there, even with a cell therapy, because it's just logistically very difficult with cell therapies. That said, cell therapies have shown fantastic promise and efficacy, to the point of almost curative in some instances, which is absolutely amazing from a medical perspective. I think that so those therapies and people developing those therapies, obviously, we need them to continue to do that sort of thing, because the potential for us to advance treating disease is huge as a result of that. And as with all new types of therapies, pricing comes down over time. The logistics are worked out, the cold chain, supply chains, etc., improve, and so access becomes easier. But yeah, I think at the moment, I certainly wouldn't think that it would be an absolute hard stop if you're coming up against a company that's developing a cell or a gene therapy. Not sure if that was quite the answer to the question, though, Derek.
That's okay. It was a long, detailed answer, and people have forgotten what the question was by now, so it's fine.
Including you and I.
But they thought of something else, right?
Yeah, no, really there.
I think you also mentioned, what about the companies producing those therapies if it is their only candidate?
Right.
I think, in those instances, it is difficult because the expense incurred very often in the development of these sorts of products is higher than it would be in, say, a standard monoclonal antibody or something like that. So it's this sort of scenario where looking at those early signals in development, really understanding where are the potentials for risk. So, for example, it's going to be around the relevance of the models that you're using, the relevance of the in vitro assessments, the relevance of the animal models if you are using a particular animal model, but also and especially understanding where the risks are associated with manufacturing and what that supply chain looks like, to me would be absolutely critical. And then the third point would be critical to that would be how you design the first in human study, what centers you're going to go to, because you're going to need to have centers that can handle these sorts of therapies. They may need to do on-site manufacture to GMP for a cell therapy, which is, not an easy thing to do. So these are the sorts of things that I would be, really keen on looking at very early, so that I'm not spending a lot of time and resource and then getting to a point where I go, oh goodness, I didn't think about that, and I'm now delayed by a year, or I can't progress.
So let's shift gears here a little bit and talk about sort of how a company, like an early stage company, how do they think about, let's take it back even one step further, right? They have some funding, they're developing their TPP, they're preparing to think about their studies. How do they think about, do they do their studies in-house? Do they have access to do that? Or do they work with a company like Lonza, right? Talk to us about what founders in this case should be thinking about through that process.
I think the rule of thumb is, from my perspective anyway, is certainly at the very early stages where you're still in sort of early discovery, if you like, do as much as you can in-house if you have the ability to do that. Collaborate with academia if you have the chance to do that, because and this is something I find really helpful, and we actually did it when I was at the biotech company. There are a lot of great academic institutions that have a lot of very, very nice equipment that it would be very difficult to purchase on your own, right?
Yeah.
And they have experts. They have great scientists who can provide assistance. So often that's a really good way of starting off. But once you get to the point where you're starting to say, actually, this thing really could have some legs, right? We've got an idea of the mechanism of action. We think that this is a druggable target, and you start to develop some potential candidates. It's at that time that you want to start thinking, well, now we need to start looking at getting some funding. And what are people going to look at? They're going to look at the track record of the people who are involved in the project, right? Because that's often what they're really interested in. Who are they buying into?
Mm-hmm.
They're going to look at the science, obviously, but they're also going to look at, what does this development pathway look like, and what does the competitive landscape look like? Yeah. And it's at the sort of later discovery stages that we at Lonza typically start engaging with clients where they've got multiple candidates, and we can help to de-risk and optimize the pathway for them from that perspective. And it's at that stage, I think the value is that you've got a partner, and whether it be Lonza or whether it be other well-known partners in the development space, it's that you have a partner and you're working with an organization that knows what it takes to get to the milestone, and the milestone is usually the IND submission, right? And that helps often to attract the next level of funding, because people are looking at data and they're saying, right, well, this element is probably de-risked. If it's on a regulatory known production platform, well, that's pretty much de-risked, right? We're not going to have to spend a lot of time and effort switching to something else. And this is especially true in the biologic space, but obviously cell therapy space as well. And that is that, if you generate a lot of data using a system that is not necessarily commercially relevant, when you switch to something that is commercially relevant, you may have to go back and retest and check
Right.
to see if the data that you've generated are still accurate, because the product attributes change. The kinetics of the product could change, glycosylation, etc. And all of that can impact the safety, the efficacy of the product. So, you're looking at that and thinking, how much rework am I going to have to do? So when I was assessing assets, it was very much a case of, who are these people in front of me that are bringing me the idea? What is their track record? And it didn't mean that you had to be, the 16th company that you've started and all of them have gone on to billion-dollar IPOs and things like that. It was just an understanding of who these people are and how passionate they are about what they do. Looking at the mechanism, understanding whether it's first in class, because that's also important. There's additional risk. You could say commercially that's fantastic, but sometimes there's additional risk with that. Is it a proven mechanism? how many other companies are working on this sort of product, et cetera, et cetera. And then very much a case of, so how, what, on what platforms are the data being generated? What are the models that are being used? How is the product being produced? In what sort of cell lines? If it's a biologic, is it being produced? Have the data produced? And therefore, how much work am I going to have to redo to progress this to IND?
Yep. So one more kind of flipping the question here, right? So thinking about where Lonza sits in the marketplace, right? How do you think about your kind of market-facing persona, if you will? How do you think about the way that the company interacts with either smaller startup companies? And I'll split those into two, right? There's a difference between $100 million Series A round company and a $5 million spin out of academia. And also with universities now who, tend to all have these networks of service providers that they can advance their own assets. So how has Lonza kind of shifted the way that you've thought about your presence, whether regionally or whatever, in terms of how you actually are facing these kind of new wave of companies?
Yeah. So yeah, great question, Derek. I think we can probably sort of in some ways split Lonza into two as well a little bit. So Lonza is a large organization. Obviously, we help clients all the way from those in late discovery through to those, we're manufacturing commercial assets for clients. And clearly, what we're seeing in the market is the same as what I'm sure every other CDMO is seeing in the market. And that is that obviously large pharma historically have been the ones taking products through to the market, but more and more, it's smaller companies that are looking to actually take products all the way through to registration.
Right.
And so, as a result, we're also modifying how we interact with clients at Lonza. So the group that I work with, Early Development Services, we are probably the earliest touch point for clients to access the Lonza network, if you like. We're just about to do what I think is a very significant expansion for us onto the East Coast. So we're opening a new Early Development Services facility in Cambridge, Massachusetts. At the moment, we only have a site in Cambridge in the UK, so we're keeping it in the Cambridges. So it's a meaningful expansion for us to increase our footprint to better service these sort of really small clients. And I think we obviously are in a situation where we understand very much that time is of the essence, but so is accuracy. So you could go very, very quickly and spend the client's money, but they get to a point where they that it hasn't been done, things haven't been done correctly, and you need to go back and rework. I know often when we talk about drug development, we think about it as being linear, right? You start in early discovery or research and development, and you end up in registering and commercial. But it's not. It's 2 steps forwards, 3 steps back very often, because that's science, right? What we certainly try and do at Lonza is leverage the expertise we have in the sort of later stages of development to really understand what's important early in development, to identify risk, and then to work with the client to help manage and mitigate that risk. So as I said earlier, we may be working with a small biotech. We may find that there maybe is a potential immunogenicity risk or a potential manufacturability risk for a particular product. But we can then say, right, based on our experience and knowledge, these risks can be managed, or it may be based on our experience and knowledge, we could progress, but it's actually going to probably be a benefit to look at one of your backup candidates that we've been working on, which may have less of a liability, and have that discussion with the client, see how much data they've generated on the backup candidates from an efficacy perspective, for example, in their in vitro models or in vivo models, and see whether that is really feasible. And if not, to work with them to design an IND enabling package that helps to mitigate that risk as much as possible. So, you know, that's broadly speaking how we would interact with smaller clients. And, you know, 95%, no, probably more, probably 98% of the clients that our group deals with, and this is true also for our I&D group, so the group that goes into GMP development, whereas we're non-GMP development, but even the early GMP development group, the significant majority of clients are actually small biotechs.
We actually had a question from the audience I think is appropriate here. And it's sort of the general thinking about, you obviously work with lots of different clients. And I think I looked it up in advance on your website. And I think something like in 2021, you all, Lonza, had worked on 780 or 800 preclinical and clinical, you know, small and large molecules, and produced something around like 250 billion capsules, right? So clearly lots of clients, lots of different clients. How do you handle confidentiality and conflict as projects become similar, even accidentally, right? So like, what is your process for ensuring that that confidentiality? But like you said, you're also using your own expertise to help guide a client as to whether they should look at a backup, or right? How do you work through all that?
Yeah, a great question, Jennifer, and not an uncommon one in this. You know, if you've been in the services industry, working at a CRO, we had exactly the same questions as we now face working at a CDMO. But the first thing I would say, and this is the answer I always give initially, is if you think about it, for a service provider, if you cannot ensure confidentiality, you're out of the business. Yeah. Right. I mean, that is an absolute fundamental. So there can't be any sharing of information, if you like. Mm-hmm. What is true though is you can't unknow what you know. And for those of us who've been in the service industry for a while, and this is one thing I would say, which, you know, I guess biotech is probably a slightly middle ground, and then large pharma is a different beast entirely. But you see so many compounds in a very, very short space of time. If I'd stayed in biotech, at the time I was there, I was working on 3 different products. In pharma, you may see, half a dozen products in 3 years. In the service industry, you're seeing 40, 50, 60 a year.
Right.
And you're seeing the data that are coming off those, obviously not the individual project team members necessarily, but as you get to project managing these sort of programs and things like that, you start to see a huge amount, and you develop experience, and so you can leverage that experience and knowledge. And you obviously don't say, well, in this compound I saw that before, but you can say that, based on our experience of 500 plus monoclonal antibodies in the last 3 years, or bispecifics, this particular aggregation pattern that we're predicting in silico is going to cause you problems. We know that. So here's some suggestions for how you might want to modify your sequence that we know have been shown to reduce the aggregation potential, as an example. And so that's, confidentiality is absolute, and we would never disclose any information from one project to another. But knowledge is something that you gain and that you leverage. And in fact, I would argue that our clients come to us expecting that we will leverage the knowledge, and a lot of small biotechs. I know I was at that stage. I wanted my service providers to share all their knowledge with me, but I didn't want them to share the knowledge of my product with anybody else. Right. How do they get the knowledge to share with me? It's because of the products that they've worked on, right? And that's the reality. We want to leverage the expertise of these organizations. But the only way that can happen is by them gathering knowledge, right, and understanding and expertise. And so I think that is the value as well in working with an organization like Lonza, that you can tap into a really, really expert group. I must say, hand on heart, in terms of the EDS, the early development services team, I am certainly the least expert amongst the entire group. A fantastic group of experts working in that space.
You want to go? I can go. So
yeah, I can. Yeah. So one thing we spend a lot of time talking about are patients and engaging patients in both the discovery process as well as for us also in the advocacy process, right? Like ensuring access to therapeutics. And in fact, last week Tuesday, a week ago, we hosted a patient engagement summit in New York, and we had several hundred people participate. So talk to us about how patients factor into the work you do for your clients, and how they have either become more important to the process. I would argue probably over the last decade, but I'm interested in your perceptions.
Yeah, no, it's a fantastic question again, Jennifer. I think patients' involvement in the drug development process that's another area that's changed enormously, even in the last decade. I think the acknowledgement that patients should be part of this process is certainly something that I've seen a shift in. So I guess one of the great examples I think of this sort of thing is when I was working in the clinical pharmacology space. We did predominantly first in human healthy volunteer studies, but we also did have what we call some hybrid studies, so hybrid designs, which were initially in healthy subjects, and then you had a group of patients that were brought onto the study as well. And it was fantastic working with the patients. We had patients who were doing their first clinical pharmacology studies, who would give talks to other patients during recruitment drives and things like that. But also, we have open days for them to come in and have a look at a clinical pharmacology unit. Many of them had never been into one. They've been into hospitals, obviously, but never into a clinical pharmacology unit. So open days for them to come and see and ask questions and understand and actually be involved in some elements of the patient protocol, that ARM protocol design, to ensure that it was okay. And we also needed to make sure that the facility was adequate in terms of the needs of the patients. So, asking them about, because they were going to need to be in the unit for a day or two overnight, to make sure that the accommodation was suitable, that we had access to specialists to come in and treat if there were any issues that we came across during the study. So I think all of that really, really is important. And I'm not sure if you're aware, but it is topical on this particular topic, that the MHRA here in the UK just this past week has published guidelines around the publication of clinical trial data, especially in the first-in-human setting, where all clinical trials, whether they are successful or not, have to publish data within 12 months. And that's publicly available. And it's done to make sure that patients are seeing data on drugs that are potentially being developed for them. And I think it's a really good step going forward to increase transparency, increase inclusion, and make sure that, when we're asking patients for informed consent, it is genuine informed consent.
Yeah. Sorry, Derek. Poor Derek, you're having your spring allergies already.
No. I coached 6 hockey games this weekend at a tournament. This has nothing to do with allergies at all. This is better than yesterday and a market improvement from Sunday. So congratulations. This is the best that we were going to get today.
So talk to us obviously about, so our ecosystem, right? So we're in the New York sort of metro area and across New York State. Lonza is investing in, as you said, you stick with the Cambridges, right? You're developing a facility in Cambridge, Massachusetts. Talk to us about how that, until you have enough to put a facility in New York, which we hope, all right, will be in the future. Talk to us about how that might impact and help New York-based companies. What does putting a facility there offer for our companies in our ecosystem?
Yeah, so I think obviously having a presence on the East Coast is really, really important in terms of how active the US East Coast is from a biotech perspective and early drug development perspective. But there's a lot of concentration of biotech on the East Coast, and I think the facility that we have on the East Coast, at the moment we have a very successful business based out of the UK, but there are time zone differences, etc., which do make things difficult for clients who are not based in Europe. Lonza, as you know, has traditionally been a fairly European-based business, although we are expanding now into the US, and this is just one of the expansions. It's obviously in my mind the most important one because it's my group, but there are several other expansions in the US as well. I think it will allow our East Coast clients and clients across the US actually to a little bit more easily interact with Lonza, gain access to our expertise, get familiar with our early service offerings. It cuts me deep when I have discussions with clients and they go, gosh, I never knew Lonza did this sort of thing. I thought you just did GMP manufacturing. And I turn to my marketing colleagues and pull what little hair I have left out, because we do a lot of marketing around this space. But I think that's the important aspect, just to get exposure to small biotech on the East Coast, and certainly from the New York area as well as from the Boston area.
And then what do you see as, because we only have a couple minutes left, if I am a CEO of an early stage company sort of in the New York ecosystem, and I'm thinking like, do I use Lonza, or what do I do? Like, how would you tell them to think about the process of selecting and working with a CDMO?
Great question. I would say that especially in the early phases, it's such a complex phase of drug development. In some ways, it's probably the most complex in terms of drug development, maybe comparable to just as you're about to get ready for a registration. Phase 2 is almost a little bit of a kind of okay, we can take a bit of a breath. But getting into first in human, there are so many different avenues of data coming at you: efficacy data, safety data, in vitro data, manufacturing data, you name it. In fact, we've got a little swim lane diagram that we've generated showing all the various parts of drug development in this phase, and how they interlink and intertwine, and it's massively complex. And so if I'm a CEO, I think probably I want to keep the lights on. I want to keep the employees that I have employed, and I want to get funding for that. That's my critical element, and progress my milestones. What I want to do is work with people who've done it before, and have the expertise and experience to help me get where I need to go, and maybe leave me to do the funding, chasing the funding, if you like, because that's critical to my role. So interact. I think I always get a good feel by working with people, speaking to people, trying to get an understanding for whether we gel as a team, if I can put it to you that way. And so I would say, if you can get on the phone, if you've got the time, and I know time is critical, but if you can get on the phone with a prospective service provider, get a feel for the people and their knowledge and understanding. Face-to-face meeting is great if possible, but it isn't always. But speak to the people in the service provider who you're looking to choose. Get that feel for their level of expertise, for how much interest they have in your product, how much they care about you as an organization, and your project. Obviously timelines are important. Price is always important, but it's more than price. It's value that's important, I think.
Yeah.
And so those are the sort of things that I would suggest.
Well, listen, that's a great way to end things. Raymond, thank you so much for joining us. You play such a critical role in the development spectrum, and you sit at really the precipice of one of the most important decisions that a biotech company can make. So thank you so much for joining us and sharing this morning. And everybody that was here, I hope you have a great day.
Thanks so much.